So welcome again to another online lecture where we are now going to talk about development and congenital malformations of the skin and its appendages. Remember that we've already looked at structural organization of the skin and its appendages in a previous lecture. And so in this particular one, we're just focusing on development and congenital malformations of the skin and its appendages. So I hope that what you know from the previous class is still there, but maybe just to guide you, we can just have a brief overview of the general organization of the skin so that when we talk about its development, you understand what is developing. So after that, we will then now focus on the embryonic origin of the skin and its appendages. We'll then talk about development of the epidermis, and we'll also talk about development of the dermis. After that, we'll pick on the glands of the skin, and in particular, talk about development of the sweat glands and that of the breast. We'll talk about some unique features about the developing skin, and we'll finish with the congenital anomalies of the integument system. So let's begin with the general organization of the skin and its appendages. We know that when we talk about the integument system in general, we are referring to the skin and its appendages. But what are these appendages of the skin? When you talk about appendage of the skin, we're referring to things like hair, nails, and various glands which are attached to the skin. We know about the sweat glands and their various types, sebaceous glands, and the way they form the pilosebaceous units, and the breast. So those are appendages of the skin. In terms of its general structure, the skin consists of two important layers. We have a layer that we call the epidermis. So the epidermis is this layer here, made up of epithelial tissue primarily. So it's a layer of epithelium. And then below that one, we have the dermis. The dermis is a connective tissue layer. That connective tissue layer is largely of fibrous connective tissue. So remember, epidermis is epithelium, and I hope you remember that it's stratified squamous keratinized epithelium. And then the dermis is fibrous connective tissue with the two parts. This part here, the papillary dermis or dermal papilla, is loose connective tissue, and the deeper part of the dermis, reticular dermis, is dense irregular connective tissue. The two parts of the layers of the skin are anchored together through these interdigitations so that we have extensions of the dermis into the epidermis. These are the ones we call the dermal papilla, and extensions of the epidermis into the dermis are the ones we call the epidermal pegs. Occasionally, you'll hear the term hypodermis. The hypodermis refers to the layer of fat just beneath the skin. It's the layer of adipose tissue below the dermis, which we generally call the subcutaneous tissue. The amount of subcutaneous tissue varies depending on body region and perhaps from one person to another. In terms of the epidermis, there are a number of cell types which are on the epidermis. That stratified squamous keratinous epithelium has a number of cell types. Keratinocytes are the majority, but we also have melanocytes, we have Langerhans cells, and you have the Merkel cells of the skin, and I hope you do remember the functions of each of those cell types. The epidermis exists in layers. It's layered from the basal side or from the deepest to superficial. We have the stratum basale or germinativum. Stratum spinosum is the largest cellular layer. Stratum granulosum is this layer of squamous cells with granules which are visible. Then you have the stratum lucidum that is best seen in glabrous skin as opposed to thin skin. and stratum corneum, which contain the dead material, that keratin layer, that the stratum corneum. Also, remember that there's something called epidermal melanin units, which refer to one melanocyte with all the keratinocytes that the melanocyte distributes its pigments to. So the concept here is that we have melanocytes usually on the stratum basale, but they have projections or processes that distribute the melanin pigment that they are produced to the various keratinocytes around them. So one melanocyte and all the keratinocytes that melanocyte is distributing its melanin to is what we call epidermal melanin units. Generally, they tend to be standard for human beings, irrespective of the race that you come from. So having given us that brief organization of the skin, we can now embark on the subject matter of the class. We start with the embryonic origin of the skin and its appendages. If you remember this particular image of the trilaminar embryo going through how that particular embryo falls into the 4th week and how the 3 layers of the gastrula differentiate, then that would be good for this particular class. Now remember, this is the ectoderm, and as time goes by, ectoderm usually undergo neurulation at the center. So the central part, which is the neuroepithelium, undergoes neurulation. After neurulation, we are going to have what we call neural tube and neural crest. The peripheral parts of the ectoderm, however, which we call surface ectoderm, will not undergo neurulation. And so that is the part we are seeing there. That's the part we are seeing there. As the embryo falls, the surface ectoderm will cover the whole embryo in a cylindrical manner, as you can see almost there in that last image there. Beneath the ectoderm is the mesoderm, which has 3 parts: paraxial mesoderm, intermediate mesoderm, and lateral plate mesoderm, which has 2 layers, the somatic and the splanchnic layer. So those are going to be important in your understanding the embryonic origin of the skin. Generally, the dermis and the epidermis have different embryonic origins. The epidermis, and in particular the keratinocytes of the epidermis, arise from surface ectoderm. So this surface ectoderm here gives rise to the epidermis of the skin. And in particular, it gives rise to the keratinocytes of the epidermis. Remember, keratinocytes are the most abundant of the cell types of the epidermis. This surface ectoderm will also give you, by extension, some glandular structures which arise from the skin, the parenchyma of the breast, the parenchyma of the sweat glands, and some parts of the hair follicle will also come from the surface ectoderm. Now, the reason is because those appendages are now epidermal derivatives. They're derivatives from the epidermis. They tend to be epidermal downgrowths. They grow from the epidermis. So we can then extend and talk about the parenchyma of the breast, the parenchyma of sweat glands arising from surface ectoderm. Then we have the mesoderm, or let me use the term mesenchyme so that it can encompass a lot of things. Mesenchymal tissue comes from, or rather gives rise to the dermis of the skin, but it also gives rise to the Langerhans cells of the epidermis. Now remember, Langerhans cells are connective tissue cells, or let me say supporting tissue cells. They are basically cells of the immune system. So they don't really come from the surface ectoderm, they come from mesenchyme, but now they invade the developing epidermis. The mesenchyme that gives rise to the dermis of the skin may have multiple sources, and we are going to talk about that shortly. So for now, understand that the dermis come from mesenchymal tissue, and we're going to talk about the origin of mesenchymal tissue of the dermis. So in this image, you can see the upper layer there being the one we are calling surface ectoderm, and the lower layer below, mesodermal, that will become mesenchymal, is the one that gives us the dermis of the skin. Apart from mesenchyme and surface ectoderm, neurocrest cells also participate in formation of the skin. In particular, the cells of neurocrest migrate and invade the developing skin. They contribute to forming the melanocytes of the skin, which are the pigment-forming cells of the skin, but they also contribute to formation of the Merkel cells of the skin. Remember, the Merkel cells of the skin are tactile receptor cells, especially their conjunction with the nerve terminals constitute what we call the Merkel's discs, which are tactile receptor cells. So that being the embryonic origin of the skin, now we can talk about the development of the two layers of the skin. Let's start with the development of the epidermis. As I've already told you, epidermis arises from the surface ectoderm. So initially, this layer is just covered by one layer of ectodermal cells, as you can see in the upper image. But as time goes by, another layer appears on top of that layer of cuboidal columnar cells, so that now that layer is represented by two layers. The epidermis is represented by two layers. An upper layer of squamous cells, which we call the periderm, and a lower layer of cuboidal columnar cells, which we call the basal layer. The basal layer functions as the region of cell division, the region where stem cells come from. So new cells come from the basal layer. And that basal layer actually stands throughout. Even in adults like in you and me, we still have that basal layer. That's the one we call the stratum basale or stratum germinativum, the layer of cell division, the layer of mitotic activity. So that layer stands throughout the whole of development and even postnatally. As time goes by, multiple layers appear because of the mitotic activity of the basal layer. The cells which have been formed from the basal layer migrate to more superficial regions. And so with the time, multiple layers will appear on the epidermis. And that explains why then the epidermis becomes stratified. So even as we look at this in the early, maybe the 3rd to the 4th week, we only have one layer. At the end of the 4th week, we have already two layers, the periderm and the basal layer. But look at what happens as we exit the embryonic period and go to the fetal period of development. We can see now the epidermis starting to become multicellular. And even as we go to the mid-fetal period or late fetal period, way into the end of the 2nd trimester, we can see that the epidermis actually is now multi-layered. So this happens progressively. The stratification of the epidermis is a progressive concept that happens. Towards term, even the keratin layer has now been formed actually. By the time the baby is being born, the epithelium is already keratinized. So understand the stratification of the epidermis is a continuous thing and occurs as a result of the basal cells dividing and migrating. So this also captures that very well. But also I want you to notice on the last image there that by this time towards term, we also now already have the neurocrest cells having migrated to the skin, and so giving rise to the melanocytes, and that melanocytes have already now developed the processes which now distribute the melanin pigment to the surrounding keratinocytes. That's what we call the epidermal melanin unit. Earlier on, we don't see those melanocytes, but they migrate and invade the epidermis, so that by the time the baby is being born, the melanocytes are already on the epidermis. Remember, they come from neurocrest. How about the dermis? So as I mentioned earlier, the dermis of the skin develops from mesenchyme that is below the surface ectoderm. In this case, this is mesoderm, which is going to give us mesenchymal tissue. But the dermis doesn't have to come from mesoderm. It may come from mesenchyme of a different embryonic origin rather than mesoderm, as we can see. But in general, remember that the dermis is a connective tissue layer. So that fibrous connective tissue layer is from mesenchyme. What are the sources of mesenchyme that give rise to the epidermis? Sorry, give rise to the dermis. We have somatic mesoderm. Remember, this is the outer layer of the lateral plate mesoderm. The somatic mesoderm gives rise to the mesenchyme that forms the skin or forms the dermis of the skin of the anterolateral body wall. So anterior and lateral body wall dermis will come from mesenchyme of somatic mesoderm origin. That would also extend to involve the dermis of the skin in the limbs. So the dermis of the skin in the limbs will also come from this particular mesoderm. This image captures for you that particular mesoderm. This is somatic mesoderm. And you can see just above it is the surface ectoderm anyway. So this somatic mesoderm gives you mesenchyme that form the dermis of the skin of the anterolateral body wall. And understand anterolateral body wall because when this embryo is going to fold, we know that this part and that part are going to fuse to form the anterior body wall anyway. How about the mesoderm here? Remember, that is what we call the paraxial mesoderm. We know that paraxial mesoderm undergoes segmentations. Cube-like segmentations of the paraxial mesoderm are the ones we call somites. Each somite divides into three or differentiates into three. We have myotome, which gives you skeletal musculature. We have sclerotome, which gives you the axial skeleton, and we have the dermatome, which gives you the dermis of the skin. Now the mesenchyme that come from dermatome or the mesenchyme that come from paraxial mesoderm will give you the dermis of the skin of the back. So basically the back skin comes from paraxial mesoderm. Lastly, we have mesenchyme, which is of neural crest origin. The mesenchyme of neural crest origin is largely involved around development of the head and neck region. And so for that reason, the mesenchyme which arise from neural crest origin contribute to formation of the dermis of the skin of the craniofacial region. So that accounts for embryonic origin of the dermis. Remember, it is mesenchymal tissue, and mesenchyme is primitive connective tissue. So that primitive connective tissue is the one that's going to form the fibrous connective tissue of the dermis, whether dermopapilar or reticular dermis. Now we can talk about the sweat glands then. Now, sweat glands develop as downgrowth from the epidermis. And these images capture for us that very well. If this is the epidermis in blue, and this is the dermis or mesenchymal tissue going to give rise to the dermis. We have some downgrowth from the epidermis. So that one, you can see what forms first is just a small bud, but that bud grows down. As it grows down, it undergoes canalization so that it acquires a lumen. Now, because it's a sweat gland, it will grow very deep and also even just coil in itself as that happens, as the canalization also continues, so that you need to have the sweat pore opening onto the skin surface. So take home message, sweat glands are downgrowth from the epidermis, which means that they are of surface ectoderm origin as well. But remember, they grow into the mesenchyme. They grow into the mesenchyme that's going to give rise to the dermis. How about breast? I'll say more about the breast. So as with regard to development of the human breast, first, the initial evidence about that band-like thickening extend from the base of the upper limb to the base of the lower limb. Okay, let me say that again. As with regard to development of the mammary gland, we have a band-like thickening of the epidermis. That band-like thickening of the epidermis extends from the base of the upper limb to the base of the lower limb, represented by this blue line. That band-like thickening is known as the milk line, or you can call it milk ridge, or mammary line, or mammary ridge. It may go with all those names. So it's a band-like thickening. Remember, a thickening of the epidermis. Now, as time goes by, in humans, that band-like thickening will involute. So many parts of it will just disappear. However, because we're humans, it doesn't disappear totally. There's a region that remains. The region that remains is usually in the anterior chest wall. Now, this is because in humans, we only have one pair of breasts, which are in the anterior chest wall. So that's the only place where the breast will form. In a particular small region, which will form the breast eventually. All right. So understand that humans have only one pair of breasts, and so we don't need the whole of the milk line. We only need the breast to be at the anterior chest wall. Now, that region that is designated to form the breast in humans will perform something. Because the milk line will be remnant on that particular region. That region is the one that's going to form the breast. What's going to happen is that the ectoderm in that region will then also undergo a downgrowth into the underlying mesenchyme. Just like the way the sweat glands developed, that's the same mechanism that also the breasts are going to develop. So in this image, you see that we have the milk line, and then the milk line disappear except the anterior chest wall. So at the anterior chest wall, that's where you're going to have the breast developing. Now, the remnant of the milk line is what we call the breast placode. That's the remnant of the milk line. And so the breast placode is only at the anterior chest wall. what happens to the breast placode? Once the breast placode has formed, this breast placode is the one that's going to now have downgrowth, so that we have proliferation of the epidermis or proliferation of ectodermal tissue towards the mesenchymal tissue as the breast develop, as you can see in those particular progressive images. So again, same concept like that of sweat glands, except that here, remember, breast is multilobulated. So it's not simple tubular gland like sweat glands. And so you try to imagine that the growth will have to take that pattern of lobulation. Maybe we go through it slowly. That once the breast placode has formed, the next thing that forms is what we call the primary breast bud. So consider this to be the breast placode. That's the breast placode. Once it has formed, the next thing is formation of what we call the primary breast bud, which is the downgrowth into the underlying mesenchyme, like that. After formation of the primary breast bud, then we have formation of secondary breast buds. What are secondary breast buds? Secondary breast buds are basically branches from the primary breast bud. So you can see they grow away from the primary bud. These are the secondary breast buds, which demarcate for you the fact that the breast is lobulated. So we expect multiple secondary breast buds. Remember, the breast has about 15 to 20 lobes. And so we expect about 15 to 20 secondary mammary buds. After the formation of the secondary mammary bud, which we can see here, remember this is the primary breast bud, that the secondary breast bud forming. After the formation of that one, as that continues, or rather, we have canalization process also going on in the ductal system. And usually the stem of the duct is the one that will begin, then extend into the various branches of the bud. So we have canalization first in what was originally the primary bud. Then we have canalization extending into what is secondary bud, as you can see in that image and also depicted in the last image there. These ones also show you the secondary buds. Then this one shows you canalization. As canalization continues, the top part, the region where we had the breast placode, goes inside. It undergoes invagination. Perhaps that could be because of the canalization process and apoptosis. So we have depression at the point of the breast placode. The depression at the point of the breast placode is what we call the mammary pit. So we have formation of the mammary pit. And that is captured here. Look at that one. That's the mammary pit, which is also captured here. That depression at the point of the breast placode is what we call the mammary pit. Now, you and I know that the breast need not to have that pit, at least postnatally. So something must happen to the pit. The pit must undergo proliferation so that it emerges out as the nipple. So this pit, eventually, there'll be proliferation of epidermal tissue here so that now the nipple everts. But right now, the nipple is inverted. You must have a stage of nipple eversion. And that shows you now a nipple that has everted, canalization has occurred, and we have lobulations as well. This image now captures for you the various stages: formation of the primary bud, formation of the secondary bud, canalization and pitting. This one shows you still canalization and pitting. And then finally, we are going to have nipple eversion as the last step. Okay. Now let's talk about some unique features of the skin that's developing. And I just want to mention two things. As the skin is developing, usually the skin has some fine hair. This hair is usually miniature, fine, and it's all over the body. We call that hair lanugo hair. As time goes by, this hair usually disappears, and it's replaced now by the permanent hair that you and I have, the one that's a bit rough. But initially, there's very fine hair that is present on the surface of the baby. We call that one lanugo hair. Now, the presence of lanugo hair may therefore mean some level of prematurity. If a baby is born and they have a lot of lanugo hair, it could allude towards possibility of prematurity. However, it is not automatic that if there is lanugo hair, then it definitely means prematurity. It's not automatic, but it may be a possibility. It may be a possibility. Apart from lanugo hair, we also have some whitish paste that may be on the skin of the fetus or the baby. This whitish paste, which is on the skin of the baby, is called vernix caseosa. Vernix caseosa is basically secretions of the sebaceous glands. So as the skin develops, you have a whitish paste on the surface of the baby, which you call vernix caseosa. It contains secretions of sebaceous gland, but may also contain some dead material as well, including hair. Now again, the presence of vernix caseosa may also allude towards prematurity, but again, it's not automatic. What we know is that vernix also disappears as the baby grows towards term. So if a baby is born prematurely, let's say 34 weeks, 32 weeks, they'll have a lot of vernix. But if they're born at term, they'll have little or no vernix. If a baby is born with vernix, it may mean prematurity, but it's not automatic that it will be premature. Great. Now we can finish with the congenital malformations of the integument system. Let's begin with this anomaly here, where we have patchy discoloration of the skin. We call this one vitiligo. Vitiligo occurs due to autoimmune destruction of the melanocytes. So generally we have patchy regions of depigmentation caused by autoimmune destruction of the melanocytes of the skin. May also have albinism. Albinism is more of a function of the melanocytes being in compromise. Usually there's an enzyme deficient, and so the type of pigment formed is not normal. So basically it's a disorder of function of melanocyte. We call that albinism. These ones are called nevi, where we have regions of heavy pigmentation. It can be very small or giant like this one. We call them melanocytic nevi, regions of heavy pigmentation. There's some regions where you may have abnormal proliferation of capillaries. We call them hemangiomas, cavernous hemangiomas of the skin. So these are abnormal proliferations of capillaries on the skin. Now, sometimes they could be part of syndrome, like the one we see in this particular image is part of a syndrome. This one we call, this picture is called port wine stain, and it could be part of a syndrome where this lady could be having also another anomaly within the brain. They are neurocutaneous disorders. May have abnormalities concerning growth of hair, like in this particular person, there's an excess hair, we call that hypertrichosis. You may also have deficient hair, which we call atrichia. Congenital atrichia is absence of hair. If you have absence of hair on the scalp, then we call that one alopecia. Alopecia is absence of hair on the scalp. Alopecia could be congenital or acquired, could be partial or total. These other images show us what we call ichthyosis. Ichthyosis due to excess keratinization. So the skin usually become very thick with excess keratin and usually show a lot of cracking, as we can see in these babies. We call this one ichthyosis, excess keratinization that lead to the cracking of the skin. You can also have anomalies that affect breast, like in this one where we have extra nipple, we call that polythelia. Polythelia is supernumerary breast, which are supernumerary nipple, which are due to persistence of milk line. So you may have milk line persisting in the regions not supposed to persist, and that will give you polythelia. You may also have polymastia if that particular breast actually developed breast tissue. So the extra breast, most of the time, are usually along the milk line still, but on the upper part, like that is an extra breast in addition to the major breast. Could be hard to tell maybe until perhaps when this lady become pregnant with the first child, that's when now they might be prominent. But before that, sometimes difficult to tell. Apart from that, we may also have congenital absence of the breast, and this is what we call amastia. This is the front part of this particular woman. And so there are no breasts, we call that amastia. It could be part of a syndrome as well. Like it would be also with congenital absence of the pectoralis major in what we call Poland syndrome. Inverted nipple is a common thing where the nipple fails to invert the last stage, remember, so the nipple remains as inverted. That's what you call inverted nipple. Lastly, you may also have breast in men, and breast in men is what we call gynecomastia, male breast. Good, so that's what I had for you in that particular class. Thank you very much.